Yeah, so we will keep a session on Friday. We will discuss everything, how you can build your connections, how you can get internships, about the projects, mini projects, your GitHub profile. So we will keep a session on this. Okay, so let's continue with our today's session. So in the previous lesson, we learned how to read a FASTA file, right? So this was the program that we wrote to read a FASTA file, and this is the FASTA file that we downloaded last time. So as we can see that this FASTA file contains only a single sequence. So we learned that whenever there is a greater than sign, it means this is a FASTA file, and this is a single sequence. But when we work with real data in bioinformatics, FASTA files often contain many sequences. For example, if I copy the sequence, okay, and paste it here. Let's paste it. Now I have two sequences. As you can see, this is sequence number 2, this is sequence number 1. Let's paste it one more time. Okay. So as you can see that now I have multiple sequences. As we discussed earlier, if there is a greater than sign, that means this is a FASTA file format. So here you can see this is another greater than sign. This means this is another sequence. Now this is another sequence. So now we have multiple sequence. We can see here as well. This is sequence 1, this is sequence 2, this is sequence 3. So now we have multiple sequences in a single FASTA file. So last time we learned how to read a FASTA file, but that FASTA file contained only a single sequence. So we learned that to read a FASTA file, we use a module called sequence IO. And sequence IO means sequence input and output. So we give an input and the Biopython give us an output. Okay. And this is how we used the sequence IO, and we called the function read. So this read function is only applicable until this FASTA file contains only a single sequence. But now at this moment, this FASTA file is now having multiple sequences. So in this case, this read function is not applicable. To read multiple sequences in a FASTA file, we call a function called parse. Okay, so if our FASTA file contain only a single sequence, so we will call the function read. But if our FASTA file contains multiple sequences, so we call a function called parse. Okay, now we will have to learn how to read a FASTA file which is containing multiple sequences. So let's open our today's lesson file, lesson 10.py. So first of all, what do we have to do? We have to import the sequence IO module in our Python file. So let's import it. So I'm writing from bio import sequence IO. Okay, so we have learned that we have to import the sequence IO module in our file. Now, as we are seeing that this file contains multiple sequences. So we want Biopython to read this file automatically. Because for example, let's have some little change. Let's add here A nucleotides. So I have added A nucleotides 3 times. Let's add here A nucleotides 5 times. Okay, so I have made a little changes. Now, as we can see, we have multiple sequences, and now all these 3 sequences are different now. All these 3 sequences have different length. Okay. So to read this file, we don't want to manually type anything. We don't want to manually read these 3 sequences. We want Biopython to do this automatically. So we have learned one thing in our previous lessons. We have learned about loop. And why we use loop? Because when we want Python to do something automatically, we use loop. So now today we are going to use loop because now this file contains multiple sequences. And we have learned about the for loop, right? So what is a for loop? That whenever we want Python to repeat the same task multiple times automatically, that's where we use a for loop. So to create a for loop, first of all, we will create a for keyword. Here we will write a for keyword. Now we have learned that after writing the 4 keyword, what do we write? We give the temporary name of a variable, right? Variable is the temporary name here. So here, for example, I write record. So here I have given the temporary variable record. And now where I want the loop to run, I want to run loop in this file. So I will write in sequence IO, and now we will call that function which is designed to read the multiple sequences in a FASTA file, and that function is parse. Okay, so here we have created a loop for record, which is the variable, in sequence IO dot parse, and we have learned whenever we want to call a function, we add dot, means dot operator, then we call our function, and at the end we add parenthesis. So inside this parenthesis, we give our input. So what input we are going to give? We are going to give the input of this file. So here, let's write our file name, which is cox1.fasta. And what else do we write? We give the file format. So our file is in the format of FASTA. And at the end, we add colon. So this is the complete syntax to create a loop. So here we have created a loop. Now press enter. Now here we will print all the information which is present in this file. We can print ID now, we can print description now. So let's print it. Let's write print the name of our variable, record, then dot ID. So here we are printing ID. Let's print record dot description. Let's print the length of our sequence. Now here I am printing dash 30 times. Okay, so why I am printing a dash 30 times? Because here we have created a loop. So inside this file we have 3 sequences, means the loop will run 3 times. So in the first time, when the loop will run first time, so all this information will get saved inside this record variable, right? So we know that Python runs from up to down, line by line. Okay, so first of all, it will print the ID, then it will print the description, then it will print the length, and then it will print the line, because I want that after every information that is going to print in the terminal, I want it to get separated. I don't want it to be messy. I want it to get separated. That's why I'm printing a line. So, okay, so that's why I have added a dash and I have multiplied it by 30. So, it will create a line. Okay, now in the second loop, what will happen? Now Python will come on to this sequence. All this information will get saved inside the variable record. Now this time Python will print me the ID, description, length, and this line of this second sequence. Okay, for example, here the sequence is the same, the ID is the same, but the length has changed now. Here I have added some nucleotides, so the length is changed now. Now in the third loop, here Python will come on the third sequence. This is our third sequence. Now the third loop will run. In the third loop, all this information, ID, description, the sequence, everything will get saved inside this record variable, and this will print us again line by line. So let's run our program once. So here in the output, we are seeing that from here till here, including this line, this was our first loop. Okay, so from here to here, this line, this was our second loop, and from here till here line, this was our third loop. So as we know, Python runs line by line. So in the first loop, Python has printed us ID. Here, let's highlight it. So here Python has printed us ID, Python has printed us description, and Python has printed us length, and then Python printed this line. Okay, then we are seeing the difference in the length. So here the length is 1542. There is difference in length here. There is 46, and then there is 1548, and then the Python printed the line. Okay, so there is the difference that if your FASTA file contains only a single sequence, so in that case, this is the program that you are going to write. This is the function you are going to call. But if your FASTA file contains multiple sequences, in that case, the read function will not work. In that case, you have to call the function parse, and there you will use loop because now you have multiple sequences. Okay, so the difference is clear. Are we clear with how to use? Yashraj, it's okay, it's fine. Okay, so are we clear how to read a FASTA file which have multiple sequences? Okay, Vidasri, what do you want me to repeat? Vidasri, can you please specify what do you want me to repeat? The last line of the program, are you asking about the line that I printed? Okay. Yes, Abhay, there is while loop as well, but for now we are only working with the for loop. Okay, there is a syntax for while loop as well. Vidasri, look at here. Here we already know that everything we write inside double quotes is considered as text, right? So here, for example, if I write A, so here I have written A. Let's run it. So see, now A is being printed 30 times. So what I am doing, I am just multiplying it by 30, that I want A to be printed 30 times. So if you will count it, this is 30 times. Okay, I am just multiplying a string. Okay, so I wanted a line because I don't want it to get messy. So I want to, for example, separate it. Okay, that's why I printed a line here. Let's rerun it again. Okay, so here the dash has been multiplied 30 times. So it's just a string. We learned that anything we write inside double quotes is a string. Okay. Vidasri, 30 is a number. It is an integer. See here, Vidasri, 30 is a number. Let me zoom. Here, see, 30 is just a number. We have learned multiplication. We have learned the simple multiplications we can do in Python, right? In the starting of the lessons. So I am just multiplying the string 30 times. 30 is just a number. Is it clear, Vedasri? Yes, Sudeshna. Yes, very good, Sudeshna. Yes, I just wanted it to get separated because we are running the loop 3 times, because in our FASTA file we have 3 sequences. So we want to see the information of those 3 sequences separately. That's why I printed the line. As we are seeing that Python runs from top to bottom, first line, then second line, then third line, and then the fourth line. Okay, so at the last, so in the first loop, in the last position, Python will print me this line, and then the second loop will run again. So in the second loop, Python will print me ID, description, record, length of the record, and then Python will print me the line, and then the third loop will run again. Okay, that's how the loop works. Is there any other doubt? Yes. Any other doubt? Okay, so let's move forward. Today we are going to work with one more file as well. So as we have now learned about FASTA file, that we learned sequences comes from experiments, they come from databases. We downloaded the FASTA file from a database as well. We downloaded the FASTA file from NCBI. Okay, so we learned that the sequences comes in the form of files, and we have learned one file format, FASTA file. So there is another file format called GenBank file. So there is a very major difference between a FASTA format and a GenBank format. Let's see our FASTA file again. So here we have already downloaded the FASTA file, as we can see. So here we are seeing that in the FASTA file, we learned there are two parts. There is this header line and then there is this sequence line, right? In the FASTA file, there is only two parts, header line and sequence line. So as you can see in the header line, there is nothing much of information about the sequence. There is only a sequence ID, there is Homo sapiens, like it belongs from humans, and there is the gene name. Okay, there is not much information about this sequence. So a GenBank file has much more information than a FASTA file. So today we are going to see how a GenBank file looks like, and today we are going to download that file as well, and we are going to read that GenBank file. So let's open our Google. Yes, Sudeshna, you can print space as well. Okay, try it, Sudeshna. Share me your screenshot, okay, how you're going to do it. Okay, Sudeshna? Yes, Ayush, very good. So let's open our Google. Search for NCBI. Here is the official website, NCBI. Okay, so let's download the GenBank format of the same gene which we have downloaded for the FASTA format. So we have selected the nucleotide here. Okay, let's copy the gene name. Okay, so this is the same gene whose FASTA format that we downloaded. Let's click on the search button. Okay, so we downloaded the FASTA format of this gene from here, right? We click on this FASTA option. So as we can see that there is GenBank option as well. So means both file formats are available of this gene. In fact, both file formats are available for every genome. So here we can download any file format that we want. So today we are working with GenBank file format. So let's click here. Okay, so this is a GenBank file format. As we can see, there is a much larger difference. There are many things written here that you can get confused about, that you don't know. We will learn it. Let's just look at the FASTA file again. Let's click here, FASTA option. And see, there is nothing information. There is not even a single information that can help us with this sequence. There is just only a sequence ID and a little description. But in the GenBank format, let's go back. Here in the GenBank format, we are seeing very large information related to the sequence. The sequence is same. This is the same gene, but we are looking it in two different file formats. So this is the GenBank format and this was the FASTA format. Now, as we learned that FASTA has two parts, a header line and a sequence line. So here GenBank file format has three parts. Okay, this GenBank file format is divided into three parts. So from here, from locus to the comment, this is called header, or we can say the general information. So here the general information is this. We have already learned about this, that this is the sequence ID, this is the length of the sequence, this is the type of the sequence, whether it's DNA, whether it's RNA, whether it's protein. Then here is the topology, whether it's linear or whether it's circular. Then there is PRI, and then there is the date. And here we can see some little description as well. Here we can see accession number. Accession number is same as the sequence ID. Okay, so there is not much a difference here. Then we are seeing some version. Version is like how many times this gene was updated. Okay, how many times this sequence was updated. For example, this sequence was updated 5 times. So here you have seen 0.5. Okay, that's how this version works. Then you have the source. So all these things we have also seen in the FASTA format. Okay, that's why this is called general information. So from locus to the comment, this is called general information. Now, this is the most important part of the GenBank file. This is the feature section. Features is the most important section of the GenBank file format because this tells us which part of the sequence is biologically important and most importantly where they are located. Their location is also available in this section. Okay, so this feature contains some annotations. Okay, now what do you mean by annotations? I know annotation is a very heavy word, but its meaning is very simple. So annotations are just extra information. So as we can see in FASTA file, nothing was written there. Okay, so we can say that there are some extra informations. So extra information is just called annotations. So as you can see, there are some slashes at the starting. You can see these slashes, right? So everything that is starting from a slash, these are called annotations. Okay, so annotations just include organism name, molecule type, database reference. We saw the reference here. Okay, here is the reference. So annotations means some extra information. So let's look in this features section, as we are saying that this feature section is the most important part of the GenBank file format. So as you can see, there are three types. There is source, there is gene, and there is this CDS. Means this feature contains three types. So there are three types of features. There is source feature, there is gene feature, and there is CDS feature. So in this source feature, as you can see, here it describes where this DNA come from, as it is showing it is come from Homo sapiens, means organism. This DNA has come from human. Now the next line, it is showing that this DNA is from mitochondria. If this DNA was from nucleus, here nucleus would have written, but this DNA has come from mitochondria, so here mitochondrion has written. Then you are seeing this CDS. This CDS is called coding sequence. So this is the part of the DNA that actually makes a protein. As you might all know, maybe some of you don't know, that the whole DNA does not make protein, right? The whole DNA does not make protein. Only this CDS is the actual part that makes the protein. So this is what written here. Now what protein it is making? Here it is written in the product annotation that the protein it is making is cytochrome C oxidase subunit 1. So this is the product, this is the protein it is making, and the gene name is COX1. So this gene is producing this protein. We can see the protein ID as well. And then here we have the translation. So here this is the protein sequence made from this DNA. So we are just getting an idea what kind of information is stored inside a GenBank file. Okay, so if we are just working lightly, we don't want much information. So for that we use FASTA files. But if we want some deeper knowledge regarding the sequence we are working, so we download the GenBank file format. Okay, and then at the last, this is the third part of this GenBank file. Here after the origin, you can see this is your DNA sequence. You are seeing some numbers here as well. These numbers are showing which nucleotide is present at which position. So we can see that A is on position number 1. Now the second line is starting from 61, means the last in this last line here A is at the last, which means A is at the position number 60. So here T is starting from position number 61. So this is showing the position of each nucleotides as well. Okay, and at the end you are seeing two slashes. This means this is the end of a GenBank file. Now we are going to download this GenBank file. Just like we downloaded the FASTA file, in the same way we are going to download this GenBank file. So here we have to go to the send to option. So click here. So complete record is already selected. You have to choose the file option again. Here GenBank format is already selected. Now you have to click on create file. Okay, so let's wait. So as we can see that our GenBank file is downloaded. Now what we have to do, we have to go to our file explorer. We have to go into the download section. Now cut this file from here and paste it in a folder. So this is the folder that I have opened inside my VS Code. So I'm going to paste this folder here, the file here. Okay, so I have pasted my, here we can see it is GenBank file format. So I have pasted this file inside my folder which I have opened in my VS Code. So let's open our VS Code again. So we can see the GenBank file format here. Okay, so let's rename it. So I'm going to name it cox1.gb. Now this is the FASTA file and this is the GenBank file. So this is the same format that we saw inside the NCBI. Now we are going to learn how to read this file. As this GenBank file contains much more information than FASTA file, so we have to learn how to read this file. So we are going to import the same sequence IO module in our file. As we have learned that sequence IO module is designed specifically to read the biological files. So that's why we are using this same module. We have written this line, means we have imported the sequence IO module inside our Python file. Now we have to print all the information, means we have to read this file. So here, as we know, now this file has only one sequence, right? So here, let's create a variable. I will create a variable called record. Now what do? We use the sequence IO, and then here we will call the function read, right? We have already understood the difference. When do we call the read function? When there is only a single sequence in our file. So here I have called the function read. Now we will add parenthesis. Now inside this parenthesis we will give our input. So what input do we give? We write the name of our file. So this time our file name is cox1.gb, and now we write the file format. So here our file format is GenBank. So this is the complete syntax that you will write to read your GenBank file. You will create a variable, you will use sequence IO module, you will call the function read, and then you will give your input of your file name and your file format. Now, we can print all the information. Write the name of your variable, record. Now we can print ID. We can print, this is the same that we printed in FASTA file as well. We can print description. Now what's new that we can print? We can print annotations. We just learned the word annotations while we were studying the GenBank file, that what are annotations? Those were the extra information. So let's print record dot annotations. So as annotations does not exist inside the FASTA file, so it will not print anything. But GenBank file contains annotations, so we can print annotations. Now let's run it. So here we are seeing that this line has printed me the ID. This is the ID. This line has printed me the description. This is the description. And this line has printed me annotations. Now these are annotations. What are the annotations? The extra information. So here it has printed me the molecule type. So it's DNA. If it was RNA sequence, here it would have written RNA. If it was protein sequence in this GenBank file, it would have printed me protein. Okay. So here our molecule type is DNA. Then here the topology is linear. Then here data file division is PRI. What do you mean by PRI? It means that the sequence belongs to a primate organism. Okay, then there is the date, and then there is the accession number, then there is the sequence version that we talked about. So here, these are all the extra information that is present inside our GenBank file. Okay, so you just have to write print record dot annotations. Now, the most important part that we talked about, the feature section, right? So here we will print the length of the features, like how many features do we have in this GenBank file. For example, this is a very short sequence that we have downloaded, so it have very less information, right? In this feature section, we have only 3 types. We have source, we have genes, we have CDS. Okay, so this will print us only 3. So let's print the length of the record dot features. So as we learned that all these things, all these attributes are present inside this GenBank file format. So this line, we have saved this line inside the variable record. So in the same way that we printed ID, so we have written record.id, in the same way that we have printed record.description. So in the same way here, we are writing record.features. But this time we are printing the length, like how many, I want to know that how many features are there in this GenBank file. So for this, we will print the length and then record dot features. So let's make this one a comment. Now run this. Okay, so now we are seeing that Python has printed me 3, that there are only 3 features inside this file. Now just as I told you that we have a very small file, there are not much extra features there, there are only 3. So here Python has only printed me 3. Now we can also print our features name which are present inside this file. So for this, we will create a loop. Here we have learned that if we want Python to do something automatically, I want Python to print me the name of all the features. These are the types of the features, right? So I want Python to print me that every type of feature that is present in this feature section, I want Python to print me their names automatically. So for that, we will use the loop. Okay, for that we will create a loop. So we already understand how do we create a loop. We use the for keyword, we write a variable name. So here let's write f, let's write the full name. So I am using feature. Where do we want to run the loop? We want to run the loop inside this record. So here let's write record dot features. Now inside the records, I want to run the loop inside the feature section. Okay, and then we add colon at the end, press enter. Now we will print features dot type, because we want to print every type of the feature that is present here. So as we already seen that there are only 3 types of features: source, gene and CDS. So here, that's why we have used a loop. We have written the variable name. Where we want to run the loop? We want to run the loop inside the record, because all this line, all this information is saved inside the variable record. That's why here we have written record. Now inside the record, where do we want to run the loop? We want to run the loop inside the feature section. So that's why we have written record.features. Now as we have learned that this variable means every time the loop will run, each time those value gets saved inside this variable. So that's why we have written print feature dot type. Now let's run this. So here in the output, we are seeing that this loop has printed me the names of every type of features that were present in those feature section. So there were source, there was gene and there was CDS. And the main point is we can also print their location, like where the source is present, in which location of the sequence, where the gene is present, in which location of the sequence. So we can print that as well. So for that, we will only here add comma and we will just write feature dot location. Okay, just like we have printed the types of the feature, similarly we are printing the location, like where they are located inside the sequence. So let's run this one more time. So here we are saying that this has printed us the type of the feature. So the first type of the feature was source and it has printed its location. So its location is this. So here this means these are the positions, that it starts from the position number 0 and it ends at the position number 1542. So as we already learned in Python that the indexing starts from 0, right? Means if I go in this, here in this sequence, in Python, the indexing starts from 0. So this means here the location of this sources, it starts at the position number 0 and it ends at the last position. Okay, in the same way, here in the same way, it's the location of this gene and it is the location of the series. So here you are seeing a symbol, but here you are not seeing a symbol. So what does this mean? So here a symbol means, for example, let's understand one thing. DNA has two strands. There is a positive strand and there is a negative strand. I will repeat again. A DNA has two strands, a positive strand and a negative strand. So here this positive means that the feature is on the forward strand of DNA. So these plus means that the DNA, here the sequence is on the positive strand of the DNA. And here the greater than symbol means that the exact starting position is not precisely known. That's why there is greater than symbol. So here its position is precisely known. That's why there is no greater than symbol and there is this plus sign, that the feature is on the forward strand of the DNA. So here this feature is also on the forward strand of the DNA and this feature is also on the forward strand of the DNA. But here is this symbol, it means its starting position is not precisely known. That's why there is this symbol. So are we clear how to read these features, how to print these annotations? So as we can see, there is much information present in the GenBank format. So this is how you read a GenBank format. It is important because we are just taking a very small example. We have a very large sequence, we have a lot of information. So for this we use programming. For this we write these codes to print, like how many features do we have. Here we printed the length of the feature, but as we are taking a very small example, we can also see from here as well that there is only three types of features: source, gene, CDS. But when we are working with large data, when we are working with long sequences, so we need this programming. We need to learn how to read a file using programming, using BioPython. So are we clear how to read a GenBank file? Okay, Aditya, what code are you talking about? Aditya, can you please specify what code are you talking about? Okay, is it clear? Okay, so there are many file formats, but FASTA format is the most common one, and the GenBank format contains the information. That's why we use these two formats mostly. Okay, so that was necessary for you to learn how to read these FASTA formats. Is it clear? You don't have any doubt? Yes, Anurag, if you have multiple sequences in a GenBank file, then you can use a loop. As we have already learned, why do we use a loop? Because we want Python to do the same thing again and again, but we don't want to do it manually. That's why we use a loop. Okay, that's why we use the loop in the FASTA file. So here, in this FASTA file, I copied and pasted the same sequence 2 times. So this made this that there are multiple sequences in this FASTA file. So here this is the necessity to create a loop to read the information of all these 3 sequences. Let's just assume that these are not the same sequences, these are different sequences. Now you need a loop to read the information regarding all these 3 sequences automatically. That's why we used the loop. But when this FASTA file only contained single sequence, let's remove these 2 sequences. Okay, now this FASTA file contain only a single sequence. So there is no need to create a loop. Okay, that's why we used the function read. But when this file contains multiple sequences, there we use loop and there we use the function parse. So if your GenBank file contains multiple sequences, you can use a loop. Okay, Vedasri, I will check it. Are we clear with today's topic? Okay, so I'm thinking of conducting a test this Friday. Are you guys comfortable? I'm thinking of conducted a, but I think you guys use AI